Okay, so chapter 6 is the same as chapter 7 in the 10th edition. Okay, so not a lot has changed. Okay, so one of the main differences between prokaryotes and eukaryotes is the way that they reproduce. So prokaryotes are simple organisms, so they don't go through mitosis. They go through what's called binary fission. So binary fission just means there are two things and they divide. So key features of binary fission is that DNA synthesis is continuous. So the cells, if they're alive, they're making DNA. Okay, all cells produced as a result of binary fission are identical. So they're all clones of each other, unless there's some sort of mutation. And we'll talk about that in the bacterial genetics chapter. The chromosomes, we only have one chromosome in bacteria, so it's attached to the cell membrane. So it's going to be a different setup. We're not going to have mitosis and all those stages of mitosis. The chromosomes are just going to divide. The chromosome is just going to divide after it's replicated. Okay, so here is the diagram of it. So what's going to happen is the cell wall and the cell membrane and the nucleus are going to get wider or elongated, and the nucleoid is the region where the chromosome is, and that's going to divide. We get a transverse septum formed. Okay, the septum becomes complete, and then we end up with two daughter cells. Okay, some organisms bud. So budding is different than binary fission. Binary fission, the cells are the same size. In budding, what happens is a small piece of something buds or breaks off. So yeast, if you looked at yeast, if you're getting ready to make bread, yeast, if you see a bud, it's going to be this smaller little piece coming off. So like here's an example. There's some budding. Okay, now whenever we culture bacteria, whenever we grow it, we want a pure culture. So we only want one thing growing in it. We need what we call medium or media to grow it. So we want something that the bacteria can eat or can use for nutrition to grow them on. And this picture just shows a wide variety of different kinds of media. And so it comes in different formats. So we can have petri dishes, we can have slants, we can have broth. There's just a endless ideas that people have of how we can actually grow bacteria. Okay, so types of media. So we have agar and we also have broth. So agar is what we used in lab this week. So it's agar is a polysaccharide. It comes from seaweed. You could think of it, so agarose, so lactose, maltose, so it's a sugar. Okay, we just call it agar when we use it for bacteria or for microbiology. So this is a picture of a petri dish, and it's got some colonies growing on it. Okay, some things that make agar special is the idea that when it's above greater than 65 degrees Celsius, it's liquid, but as it cools to room temperature, it turns solid. So we can put it in an autoclave and heat it up, and it'll be liquid, and we can pour it into whatever format we want. So we could pour it into petri plates, we could put it in tubes, so we can have the tubes be just level, okay, so then you would stab down into it, or we can make slants. So a slant, what you do is you pour the media into it and you tilt it. So this is a picture of a whole bunch of slants and bacteria growing on the surface of the slant. Okay, so why do we use agar? So we use agar because it's got these distinct properties. So one, right, is that it's greater than 65 degrees Celsius. It's liquid, but then it forms a solid as it cools. Okay, the other thing is the bacteria don't break it down. So microorganisms can't digest it. So it can't be broken down. Okay, we want a surface for the organisms to grow on. If we have a broth, the broth tends to get cloudy and you can't see individual colonies, but with agar we can see individual colonies. So all of this kind of goes together. Okay, so broths stay liquid, but we can get a lot of stuff to grow in there. We can get tons of things, millions or billions of bacteria to grow in it. So we use them because they're very nutrient rich. We can get lots of stuff growing in it. We can shake them and introduce oxygen to them. Okay, now if we are growing something that's living, well, wait a minute, let me start over again. I'm getting distracted, sorry. If we are growing viruses, okay, viruses need a living host. So we grow viruses on things like eggs or animal cells or animals. So animal cell culture is really interesting. So it has, they take cells or they take an organ from an animal. So say this little hamster, they take the ovaries out of the hamster. Okay, and they can dissect out the ovaries and they can put it into a broth that has the right nutrients, and those cells can grow. Okay, cell culture is where a lot of research is going on. It's how we make a lot of vaccines. It's how we make monoclonal antibodies. A lot of drugs actually come from cell culture. Cell culture has an interesting history. One of the ways, the way it was started in the first human cells that were cultured that could reproduce forever are called HeLa cells. And I don't know if you've actually heard of them before, but I'm going to put a link to HeLa cells on the Moodle page so you guys can read about it, because it's a very interesting story. Okay, so now when you go back to talk about media and the different kinds of media that we have, okay, some are synthetic, so they're prepared from materials in the lab. Some are very, very defined. Okay, so defined means we know exactly what's going into them. So it's really a laundry list of chemicals that are going into them. Complex, those are not very well defined. Okay, so those are things like we might use sheep's blood. So we don't know what's actually in the blood of the sheep, but we know the bacteria like to grow on it. Potato dextrose agar is another example. So you take potatoes and you just take sugar and you kind of make mashed potatoes and add agar to it. Okay, so things that are commonly found in media are peptone. So peptone are proteins that have been digested with enzymes. So the bacteria can't, they don't have a digestive tract, right? So they can't break down complex molecules inside. So lots of times we feed them things that are easy for them to absorb and easy for them to use. So peptone is digested protein. Yeast extract is yeast that's kind of been broken down. Casein is actually milk protein that's been broken down. We also give them serum from blood. So serum is the liquid portion of blood. And sometimes we give them straight up blood, and that's called blood agar. Okay, so we give them sheep's blood in the agar. Okay, now different media is used for different purposes. So we have selective, we have differential, and then we have enrichment. Okay, so back in the day, so like in the late 1800s or the early 1900s, they didn't have the diagnostic tools that we have now to identify bacteria. So what they did is they developed a bunch of different media that could show how the organisms, they're going to look different when they're growing on different things. So selective media encourages the growth of some bacteria, but it inhibits others. Okay, differential has there's something in the media that allows a change to occur that you can actually see when a certain organism is actually growing. Okay, and then some media can be differential and selective. So selective means one thing will grow but the other one won't. Differential means one thing will grow and it might look different, like the agar might turn yellow if it's a certain thing. Okay, so here's an example. So one example is called McConkey agar. So McConkey agar has crystal violet and biosalts in it. The crystal violet and the biosalts keep gram-positive bacteria from growing, but gram-negative can grow. Okay, so gram-negative can grow. So gram-negative organisms, a lot of them are found in your gut. So McConkey agar contains lactose and a pH indicator. Okay, so if the organism can use lactose, it'll ferment it. And when it ferments it, it's going to make things acidic. Okay, and the media will turn red. Okay, so things that can't use lactose are going to stay colorless. So McConkey agar is a way to take a culture from somebody's gut and then figure out what organisms are in there. Okay, so here's a picture. So colonies of a lactose fermenter are in A, they're yellow. Oh, they're red. Sorry, I said red. They should be red. Okay, and the ones that can't digest, they're just going to stay yellow. They're going to stay normal. Okay, here's another example, this hectone agar. So only gram-negatives will grow. Things that can ferment lactose are going to turn orange. Things that can produce hydrogen sulfide will turn black. So this is what they'll use to differentiate between Shigella and Salmonella if they're trying to figure out what kind of diarrhea somebody has. So the Shigella will look green, but the Salmonella will look black. Okay, now in the book there is a list of selective and differential media, and I'm not going to require you to memorize those. That's not necessary, but it's interesting to see what's out there. We'll use, let's see, we will use, not McConkey agar, shoot. Oh well, when we get to the end of the semester, sorry, I'm doing this at the end of the day and my brain's not working. We'll use some differential and selective media at the end of the semester in lab. I don't have it. Okay, now enrichment media. Some organisms are what we call fastidious, so they're really hard to grow. They grow well in the body because they're getting nutrients from the body, or they grow well in a plant because they're getting certain nutrients from the plant. But they're hard to grow in a culture. So enrichment media means we've added something special to the media so that the bacteria can grow. So we say special nutrients added for fussy eaters. So this is sheep's blood agar. And then this is actually, they call it chocolate agar, but it's sheep's blood agar. So it's blood that's actually been autoclaved, so the blood's been cooked. So they say it looks like milk chocolate, which is really gross. But bacteria love it. Everything will grow on it. It's like having macaroni and cheese or something for organisms. So that's enrichment media. Okay, now I'm going to stop here because this is right before the next set of PowerPoints. So whenever we're trying to grow things, we usually want a single colony. We want to be able to have things separated out so we can actually see them. So a CFU is a colony forming unit. And a colony forming unit is what gives rise to a colony. So there's several different ways of getting these colonies, and that's what we're going to talk about next.